Anil Dhawan requires no introduction. He is a well-known personality, and most of us, or nearly all of us, would know him. Usually, you would have listened to his talk about acute liver failure or hepatocyte transplant. For a change, today he is going to talk on gut microbiome and liver. Professor Anil Dhawan is my mentor and he has trained me. And he is the director for Pediatric Liver, GI and Nutrition Center at King's College, London. And his core area is research in hepatocyte transplantation. Over to you, Professor Dhawan. Please enlighten us.
Ladies and gentlemen, colleagues, and everybody in the audience, ISPGHAN, thank you for inviting me again. The topic I'm asked to speak on is gut microbiota and liver disease. I think the knowledge of gut microbiota is not recent. It goes up to 400 BC when Hippocrates, under his statements, one of the statements he made about gut microbiota, he felt, or he conveyed to us, that gut microbiota is a forgotten organ. And what he wanted to say is that the microbes in the intestinal system, which are majority of our microbes, are the cause of where all the disease begins in the gut, death sits in the bowels, and bad digestion is the root of all evil. You may have heard people saying these things in a lighter vein, that everything starts from the stomach ache or belly not well, etc. But the reason that we haven't made much progress over 400 years is that our understanding of gut microbe or microbiome was based on microbiological means identification on the culture. And the culture methods has been very basic. And so there are so many species and genera, as I will show you later, that it has been difficult to identify all of them on microbiological culture standard methods. So till such time when we had genomic sequencing, the field has really expanded. And what you look at here in this slide is that in the last 2 decades, there's an exponential increase in the number of publications on this subject. And between 2013, 2017, there's something like 13,000 publications on the subject. What caught my attention about 9 years back when we started work on this field was a full article on The Economist, and it carried a lot of information which could be understood by a layperson and emphasized that how important the gut microbiome is, and most of our non-communicable illnesses have a genesis in gut microbiome. So that's when we started our interest at King's College Hospital, and Vandana Jain, who was one of the MD students, she started her MD on this now, and she nearly completed. The data is still to be analyzed. I will show you later. Just some basics. To understand gut microbiota, you imagine a rainforest, okay? So rainforest, or a forest which has all kind of different trees, different shrubs, all sizes, all colors, different quantities. Some areas of the forest are rich in one particular type, and others are other species of trees. But by and large, the whole forest has an equilibrium of these trees or these shrubs. There are 100 trillion bacteria versus 10 trillion cells in the body. So our number of bacteria is 1 in 10. So for every 1 human cell, there are 10 bacteria. Okay, there are 40,000 bacterial species and maybe more, and there are 9 million unique bacterial genes, which is more than 150 times of our genome gene. So what it tells us is it's a massive tissue mass of bacteria in our gut, and majority of it sits in the gut. It's about 2 to 3 kilograms of our body weight. Now, if you look at the gut changes from stomach to small intestine to large intestine, and most of the microbiome is in the gut, 90%. Stomach and duodenum, the most common family we found are lactobacilli and streptococci. As you move to the small intestine, jejunum, ileum, the number of bacterial count increases from 10 raised to 1 to 10 raised to 4 to 10 raised to 8 colony forming unit per mil. And lactobacilli subsequently get diluted by coliforms and Bacteroides. As we go into the colon, bacteroids become the most important organism, followed by bifidobacteria. Now, this is a hierarchy of taxonomical classification. Now, it tells you that, you remember we read in the biology, there's a domain, kingdom, phylum, family, class, order, genus, and species. I think what we need to know is a little bit about the phylum. And what we need to understand is that most common phyla that we look after, or that we are interested in the liver and gastrointestinal axis, is Firmicutes, which are 60 to 80%, Bacteroides 20 to 30, Actinobacteria, and Proteobacteria, in that order. Now, these are the most common one, and the organisms that are of relevance to us in the field of cirrhosis, NAFLD, or sclerosing cholangitis, biliary atresia are these fives: Bacteroides, Actinobacteria, Firmicutes, Proteobacteria, and Fusobacteria, and further of their families. So, as I said already, there hasn't been much progress because of lack of technology. It's only in the last 2 decades we have got access to sequencing, and previously all the bacterial studies were done on culture. So now we are talking of next generation sequencers. So stool sample bacteria DNA is extracted, and 16S RNA amplification is done by PCR, and then you're looking at RNA database, in our database, compare it and identify the organisms. As I already said, only 30% of the microbiota are culturable. That means if you just rely on a culture technique, then 70% will be missed. What is relevant gut-liver axis? I think so far what we know is, in the literature, fatty liver disease has attracted a lot of attention, then any cirrhosis, and then we talk about biliary atresia a little bit in this talk. And gut-liver axis, the gut is attracted to, connected to liver. So if you look at here, the portal venous system is the entry from the gut to the liver. And most of the nutrients, toxins, they go from the gut epithelium, lumen, to the portal circulation and into the hepatic circulation, and ultimately becomes a venous circulation. Now, if you got to give a bit of a terminology, so when there is a problem with the gut microbiome, we call it gut dysbiosis. What it means is the microbiome has lost its diversity. So if you look at this picture, this is very colorful: red, yellow, green, violet, lilac, and different kinds of plants are there in this patch. If you look on the right-hand side, what you find here is there are few plants, okay? There's a conifer, there is a shrub, there is maybe a algae, but these are the very few plants you can count. And most of it is 1, 2, 3, 4 you can count. Here, there are so many species. So there is this side of flora is very diverse. So in the gut biome, gut flora, when it's healthy, there are about hundreds of microbial phyla you will find. When the gut is not healthy, the microbiome is not healthy, the diversity is in tens. So 10s, 20, 30, 40, but here you have hundreds. So increased diversity is generally a good indicator of a healthy gut. So that's what we need to remember. More diverse the gut microflora is, the healthier your system is. But gut microflora changes with age. So it's important that when the baby is in the womb, the gut microbiome is in the pregnant mother is a sterile gastrointestinal tract. At birth, depending on the mode of birth, cesarean versus vaginal delivery, different bacteria colonize. Infancy, first month, 6 months, 2 years, it changes from enterobacteria to bifidobacteria and firmicutes. Adulthood, in elderly particularly, the bacteroids take over and firmicutes go down. Not only this, the age, but the variability in gut microbiome is also dependent on geographical location, region-specific diets. Some diets are harmful, some are not. Host genetics, epigenetics, it's extremely important. Antibiotics, feeding types. So hence, the studies on gut microbiome becomes very difficult. So if you are looking at matching somebody for geographical location, what diet they eat, what the host genomics are, whether they had antibiotics or feeding type they had, were they on prebiotics or probiotics, what age they are, mode of delivery, can you imagine how many factors they are there to be taken into account? But what we do know is that if your gut microbiome is not diverse, that means it has not all kind of bacteria, then it's thin. And if it's not fiber rich, then your microbe could start eating you. So means this is a photomicrograph of a gut that has got a very diverse microbial flora, while this is very thin, and you can see how the membrane is getting affected. It's a fiber deficient diet, and we come to fiber. The short chain fatty acid that are produced by the fiber produce butyrate, and butyrate conserves the integrity of the gut permeability. A lot of work has been done on fatty liver disease, and there is a two-hit hypothesis that there is a accumulation of lipids in the hepatocyte, but you need a second hit, which is an inflammatory process that leads to hepatocellular injury. And the second hit sometimes is attributed to gut microbiome and its translocation. So if you look at this photomicrograph, what it shows is that if we have intestinal dysbiosis, as I said, change in diversity, okay, what it does is by various mechanisms, which will come later on, it releases what you call damage associated proteins, and it's a pathogen associated patterns, and what they do is they cause change the intestinal permeability. Hence your bacterial products easily find into the blood circulation, and what you call it bacterial translocation, and that switches on TLR4 receptors. It's a lipopolysaccharide binding protein that binds into the protein. Lipopolysaccharides bind to liposaccharide binding protein in the hepatocyte and upregulates TLR4, CD4 upregulation, leading to inflammation, means inflammation, and leading to inflammation, and subsequently that leads to cell death and fibrosis. So what we understand is in fatty liver disease particularly, there is a lot of information that gut microbiome changes the host energy metabolism, it increases insulin resistance, and there are several metabolites, choline metabolism, methylamines, etc. get changed, and these are generation of this hepatotoxic compounds causing liver injury. So it has been further shown that there is element of gut dysbiosis, further leads to small intestinal bacterial overgrowth, gut barrier dysfunction, and modulation of immune balance. These are the elegant studies. And if you look at obese mouse, a germ-free mouse, and germ-free mouse with, if you transplant the germ-free with microbiota from a conventional mice, they start gaining weight. And if they're fed high-fat, high-calorie diet, their weight gain is towards fatty liver disease. And if in this elegant experiment, when the germ-free mouse had a high figure, high shoot content, then you made into a converts R, we called it. But if this was transplanted a microbiome from a fatty liver disease patient, then this microbiota transfer in the germ-free mice leads to steatosis and fatty change in the liver of this mouse. Particularly, you also got insulin resistant and body fat changes here. So this is an elegant experiment. The transplantation of a microbiome from a fatty liver disease patient into a germ-free mouse creates a phenotype of fatty liver disease in the germ-free mouse. So what has been done in children? There are very few studies, and they show that in children with fatty liver disease, you have increased bacteroides and firmicutes and decreased firmicutes in NASH. So there is a signature of this bacteria or this product in patients with variable degree of fatty liver disease. Another study showed lactobacillus increase and firmicutes both increased. But what you have to remember is when you speak to people who work really in the field of microbiome, for them, the numbers are extremely important, not like us, 5 patient, 10 patient. They want hundreds and thousands of patients, which we can't do. In this study, in a biopsy-proven cohort of 86 patients, in 72 patients, there was a mild to moderate fatty liver disease. They have an increase in firmicutes species, but when they were advanced liver disease, they had gram-negative bacteria, proteobacteria. So there was a change in the microbiome of the severity of liver disease. The problem with the gut dysbiosis with NAFLD in children, the numbers are very small, but what is known is there's an increase in the file number of bacteroidetes and decrease in the firmicutes. And that's a phenomena that we all observed. In terms of treatment, people have tried probiotics, prebiotics, even fecal microbiota transplantation, but given the numbers are small and there are so many variables that I showed you earlier, hence results are really not that conclusive. Just to bridge in between, before I bring in cirrhosis, biliary atresia, there are a lot of studies that have been performed in cirrhosis patients for gut microbiome, and there are so many studies. And if you say, Jasmohan Bajaj has done enormous amount of work in this field, and what they show is there is a pattern that you can recognize, that there is an increase in proteobacteria and there is a decrease in the bacteroides, which we saw was a signature for cirrhosis. And in cirrhosis also, it depends whether you are decompensated cirrhosis, hepatic encephalopathy. If you have hepatic encephalopathy versus no encephalopathy, you have a different microbiome. What I wanted to touch base you a little bit, which we are interested is in biliary atresia. This was a editorial earlier on this year, which we try to bring the concept of how the gut microbiome could be changing the outcome of biliary atresia. We all know that biliary atresia is a condition of the obliteration of the biliary tree. We still do not know precisely the cause of this condition, but we do know that only 60% of children will clear their jaundice, and majority of them require a liver transplant within the first 2 to 3 years of life. Now, if you look at here, what we do know is there is a lot of inflammation and fibrosis, and there is this intestinal connection direct. One is the portal venous connection, but there is another intestinal connection that has been made to the biliary tree with the intestine, is the Kasai portal enterostomy, and that is slightly different than adult models where they only have this connection and not this one. So there are several factors that are seen as maybe they change the phenotype, like steroids, choleretics, antibiotics. But despite that, I think our outcomes has been consistently quite disappointing. So the hypothesis is, are there any modifiable factors like gut microbiome? If you look at in this diagram, what we try to elucidate here in this review in Journal of JPGN, is that if you look at here, normally what you get is a portal vein flow in a healthy state, your gut intact, gut barrier, so you don't get activation from this bacteria, and it's in balance. But when you have a bacterial dysbiosis, or because of the breach or the liver disease, what you get is this membrane starts disappearing and decrease in butyrate that maintains the gut permeability. Hence, the bile which was coming here can be damaging, and this microbiome can start entering into the circulation from here to the portal venous system. What you find here in this diagram, lipopolysaccharides can upregulate inflammation, while another conduit, with a biliary-enteric conduit, can also directly give you this LPS dosage or LPS injury, and causes injury to the hepatocytes. So we set out to study, is the gut microbiome associated in biliary atresia? We have a prospective study with age-matched healthy controls and disease controls. So follow-up is about 1 year up to a transplantation. So what we saw in this preliminary data is in 17 children, babies who had biliary atresia at first week, what we found is that this was their microbiome, but I'll give you a bit more details here. What you find here is these are the healthy controls, which are not, these are biliary atresia infants, and these are disease, other cholestasis children. They were 100% were exclusively breastfed the healthy controls, while biliary atresia and disease controls were not exclusively breastfed. But what you do find is that there is a pattern is different. There is a more blue here, which is bifidobacterium, but the blue here is get changed by yellow, which is streptococcus and enterococcus others. So you see a spectrum here that the gut microbiome of diseased infants is different than the healthy breastfed infants. If you look at this scatter diagram, which elegantly shows that the babies who had a normal controls, they are in red, and they are kind of a clustering up there, while the babies with biliary atresia and other liver disease also clustering on the left lower hand side. So that means they're already differentiating in terms of bacterial species they are harboring, like bifidobacterium, streptococcus, clostridium. So these are what we think are causative, contributing to the injury to the hepatocytes. If you look at it further, if you look at these two photomicrographs show the 10 most common abundant genera on the top and in the bottom, but you see there is a change over 3 months to 6 months. This is just the data on the biliary atresia patients. You see the change over the 6 months, that how the picture changes. So less bifidobacteria, there's increased biodiversity, increased pathobionts, but that's what we are observing in these pictures. So whether this is an effect of surgery, antibiotics, MCT, steroids, choleretics, I think we need a much bigger numbers to differentiate between all these factors. So to conclude, ladies and gentlemen, current data suggests that microbiome dysbiosis is related definitely in fatty liver disease. And because of disruption to intestinal epithelial barriers, translocation of pro-inflammatory substances, then it also alters the host energy metabolism by metabolite like choline and bile acid metabolism, leading to production of hepatotoxic molecules. In biliary atresia, the data is limited. I think there are very few studies. Ours will be the first relatively big prospective study. There is a study on 6 children from Holland, and there are 2-3 studies from China. Again, a small number, but not done in a prospective manner. That's what we are doing. What we have demonstrated so far in this study is that there is a less bifidobacteria, there's increased diversity, increased pathobionts, enterococcus and clostridia. We are looking at correlating this with bacterial metabolites, immunological markers in these things, and looking at jaundice-free survival versus jaundice survival, and ultimately liver transplant and outcome after that. The study is in progress, still it's not completed yet, and I hope I'll get another opportunity to share the data with you. I thank you all for listening, and I'm really happy to take some questions.
Thank you, Professor Dhawan, for this excellent talk. Thank you.